The genetic information in a bacterial cell is stored in a form of double-stranded, covalently closed circular DNA known as plasmids. Replication begins at a specific site called the origin of replication, or ORIC in case of E. coli. The origin replicates and then DNA replication proceeds into two different directions. The two original strands shown as solid lines serve as the templates for the synthesis of new strands known as dotted lines. This is referred as semi-conservative replication. The replication of the DNA begins at a sequence of nucleotides called the origin of replication. This is the origin of replication where the DNA polymer has been bound. So first the helicase unwinds the double-stranded DNA helix and single-stranded binding proteins next with the single strand they will bind to it, and that will open a bubble. The DNA polymerase III is a major enzyme involved in a DNA replication. The DNA polymerase III can only add a nucleotide to the 3' end of the pre-existing chain of the nucleotide. It cannot initiate the nucleotide reaction itself. It can only extend. Therefore, an RNA polymerase known as primase, this starts to add RNA primers at the sequence of about 10 nucleotides complementary to the parent DNA. The DNA polymerase III can then add nucleotides to the synthesized new complementary strand of the DNA. And this is how the DNA polymerase III extends the primer. Because the two parent strands of the DNA are anti-parallel, they are oriented in opposite directions and must therefore be separated with different mechanism. The leading strand elongates towards the replication fork by adding nucleotides continuously to as its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously and a series of short fragments called Okazaki fragments are generated. When the DNA polymerase III reaches the RNA primer to the lagging strand, it replaces the primer with RNA polymerase I with a deoxyribonucleotide copies. The DNA ligase then attaches the nick that is formed between the phosphodiester bond. The DNA is further unwound, new primers are added, and the DNA polymerase III jumps ahead to begin the synthesizing another Okazaki fragment. This process continues as the fork moves from 5' to the 3' direction of the existing parental DNA. For simplicity, the DNA polymerase III has been depicted here as two separate units, one acting on the leading and one acting on the lagging strand. But in reality, both the subunits are linked together with the tau subunit, which a DNA structure forms. This is the looping of the structure that is very necessary to form in order to explain how the process works simultaneously in both the strands altogether. Although mistakes can happen during DNA replication, they are extraordinarily rare. A key reason for this is proofreading function of the DNA polymerase enzyme itself. Here we'll see roughly 1 out of every 100,000 to 1 million bases attached to the 3' end of a growing DNA chain will have a mismatch, improperly base pairing with the complementary strand. When such mismatch occur, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the free primer and with the attached base to enter a 3' exonuclease site of the DNA polymerase. The exonuclease site catalyzes the removal of the several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown in this particular picture. Although multiple nucleotides are cleaved from this 3' end, following the removal of 3' to 5' direction, the DNA polymerase can then resume its synthesis activity from 5' to 3' direction. This proofreading mechanism reduces the error rate as low as 1 in 100 million for DNA polymerase III enzyme.